Hi, so we will be starting this session by covering this competency, which is on chemistry of carbohydrates. So these are the learning objectives for this session. So you should be able to classify the carbohydrates and give examples of each. Classify polysaccharides with examples. List the components of mucopolysaccharides and mention some examples. And what are the various functions of mucopolysaccharides? Okay, fine. Let's see one by one. See, why do we need to learn about carbohydrates? See, in India, this disease called diabetes is a growing challenge, with nearly 8.7% of diabetic population in the age group of 20 and 70 years. This is as per WHO data. Okay, it's a growing public health problem. So, what is wrong in patients with diabetes? Patients with diabetes have increased levels of glucose in their blood, which is nothing but a carbohydrate. So, hyperglycemia is a characteristic hallmark feature of diabetes mellitus. India leads the world with largest number of diabetic patients. So thus the knowledge of metabolism of carbohydrates and their regulation is very, very important. Carbohydrates, which is also called as saccharides, are essential and abundant class of biological molecules. The name carbohydrate literally means hydrates of carbon. Okay. The empirical formula to represent carbohydrate is CH2ON, where N is actually greater than or equal to 3. Okay, fine. So carbohydrates are polyhydric alcohols or polyols. That is, they contain numerous or multiple alcohol groups. Okay, they either contain aldehyde as functional group or ketone as a functional group. If they contain aldehyde as functional group, then they are called as aldose. If they contain ketone as a functional group, then they are called as ketose. For example, glucose is an aldose, fructose is a ketose. Okay, so this is how carbohydrates can be classified. They can be classified into monosaccharides, disaccharides, oligosaccharides, and polysaccharides. What are monosaccharides? Monosaccharides are the simplest form of carbohydrates. That is, they cannot be hydrolyzed further and are fully soluble in water. Okay, the monosaccharides can be further classified based upon the number of carbon atoms they carry and based upon the functional group they have. For example, this is how they can be classified. If the number of carbon atoms is 3, then they are called as trioses. If it is 4, then it is a tetrose. If it is a 5, it is a pentose. If it is a 6, it is hexose. And if it is a 7, it is heptose. For example, these are the trioses: glyceraldehyde and dihydroxyacetone. The only difference between these two is the functional group. See, in glyceraldehyde, it is aldehyde group. Whereas in dihydroxyacetone, it is a keto group. So, this is an aldo triose and this is a keto triose. Okay. Why do I want to know about these names? These glyceraldehyde and this dihydroxyacetone are intermediates of a pathway called glycolysis. This is the first metabolic pathway for glucose. Okay, fine. Next, what are tetroses? Tetroses are 4 carbon containing monosaccharides. Okay, fine. Here, if you see here, erythrose 1, 2, 3, 4. Again, it's an aldehyde, so it's an aldotetrose. This is erythrulose. Okay, so the name ULOSC itself suggests that it is a keto group containing sugar. Okay, again, these two are intermediates of a pathway called HMP shunt pathway, which is also called as pentose phosphate pathway. These pathways will be discussed in detail when we see about metabolism of glucose. Okay, fine. Now let's see pentoses and hexoses. Pentoses, as the name suggests, it contains 5 carbon atoms. 1, 2, 3, 4, 5. Again, it's a aldehyde containing sugar, so it's a ribose. So it's a ketose. 1, 2, 3, 4, 5. This is called ribulose. So both ribose and ribulose are also intermediates of a pathway called HMP shunt. Okay. Now let's see the hexoses. Hexoses, the name suggests, contain 6 carbon atoms. 1, 2, 3, 4, 5, 6. And this is glucose. Galactose is also an aldohexose that also contains 6 carbon atoms. Fructose is a ketohexose. It contains ketone group as a functional group. Okay, it is also a hexose. Okay, fine. Now let's see about glucose. Glucose is the most important monosaccharide. Okay, carbohydrates in the diet which we take are mostly hydrolyzed to glucose and then they are absorbed into the blood circulation. Okay, glucose is a major metabolic fuel for all the mammals. Okay, it is considered as a primary fuel for the fetus. For the growing fetus, glucose is the primary fuel. And these glucose and sugars exhibit various types of isomerism. Okay, that for example, Here there is one isomerism here. This is called DL isomerism. Okay, they are nothing but mirror images. Okay, just concentrate on the 5th carbon position. That is a carbon near to the 6th position. Okay, so based upon the orientation of the hydrogen and hydroxyl groups around the 5th carbon position, there is D and L isomer. For example, if the hydroxyl group is on the left side, that is a L glucose. If the hydroxyl group is on the right side, then that is D glucose. Okay, monosaccharides in nature mostly exist as D sugars. That is, glucose mostly exists as D glucose. Okay, fine. So why do you want to know about this D and L isomerism? How it is going to be helpful as a doctor to me? Okay, the presence of asymmetric carbon atom confers optical property to a compound. That is, ability to rotate the beam of plane polarized light. Okay, it can be either dextrorotatory or levorotatory. Okay, you would have seen these bottles in the hospitals, in the wards. Okay, they're nothing but dextrose. Dextrose is nothing but glucose. Okay, then why it's called as dextrose? Because when you take glucose and put it into a solution, because of the optical property confirmed by the asymmetric carbon, glucose will rotate the plane polarized rays towards left side, towards right side. That is, glucose exists as a dextrorotatory form. Hence, they are called as dextrose. Okay, so that's the reason why we call glucose as dextrose infusions. Okay, fine. Because glucose exists in dextrorotatory form. Next type of isomerism is called epimerism. What is epimerism? Epimerism is variation in the configuration of hydroxyl group and hydrogen group on carbon atoms 2, 3 and 4 of glucose. Okay, if the position of the hydroxyl atom varies between the two sugars around C2, C3 and C4, then they are called as epimers. Okay, for example, this is D-glucose. If you number the carbon atoms like this, C1, C2, C3, C4, C5, C6. Okay, now see D-galactose and D-mannose. Okay, they are also hexose. Just see the difference between these two. 1, 2, 3, 4, 5, 6. Okay, where is the difference? Difference is in the 4th carbon. See, the hydroxyl and OH group of glucose is different from galactose in the C4. So glucose and galactose are nothing but epimers by differing in the orientation of H and OH around 4th carbon atom. Okay, now let's look at the mannose. Where is the difference here? The difference is at the 2nd carbon atom. See here, hydroxyl group is on the left side, but here is on the right side. Okay, that's it. So glucose and mannose are also epimers with the position changing at C2. That's it. These are epimers. Epimer is due to the difference in between the position of H and OH around 2nd carbon or 3rd carbon or 4th carbon. Okay, fine. That's about epimerism. Okay, fine. Now, what are anomerism? Anomerism is variations in the configuration of hydroxyl group on an anomeric carbon. Okay, it can be either an alpha anomer or it can be either a beta anomer. Okay, so for example, glucose or any sugars, when you put into a solution, they will not exist in the linear form. Okay, they will adopt a closed form, which can be either a chair form. Okay, see here. So this is C1, C2, C3, C4, C5. Okay, the hydroxyl group at the 5th carbon will form a bond with the 1st carbon. Okay, adopting a closed conformation like this. Okay, this molecule is nothing but glucose. It is also called as alpha D glucopyranose. Okay, whereas this is called beta D glucopyranose. So what is the difference here? You have to check for, you can label a substance as alpha or beta anomers by looking at the position of hydroxyl group at C1 and at C6. Okay, here if they both exist on the same side, if the hydroxyl group at the 6th carbon and the hydroxyl group at the 1st carbon exist on the same side like this. Okay, like in a cis form. What is cis form? Cis form is what is cis and trans? Cis is drawn like this. Okay, this is cis. Okay, what is trans? This is trans. Okay, if this hydroxyl group at the 1st carbon and the hydroxyl group at the 6th carbon exist on the same side, that is in the cis form, then they are called as beta D glucopyranose. Okay, however, if they exist on the opposite sides, for example, here the OH group is on the below the ring and the C6th OH group in the C6 position is on the other side. that is, it exists in trans form. Okay, this is called alpha D anomers. Okay, so this is the difference. Okay, trans and cis. Okay, if they exist on the same side, then that is beta. If they exist on the opposite side, then it is alpha. Okay, fine. So this type of difference in structure is called Isomerism. Okay, fine. So that's about glucose. Okay, now let's see about fructose and galactose. Okay, fructose is also a monosaccharide. It is mainly present in the honey sugar or cane sugar. What are the major sources of fructose? Fruit juices, honey, sugar sweetened beverages, for example, Coke, Pepsi, etc. Excess intake of fructose is not good because it can lead to weight gain because it can increase the chance of development of fatty liver. Okay. Then galactose. Galactose is the major component of dietary product. It is one of the component of lactose. Lactose is a disaccharide. It is made up of glucose and galactose. Galactose is found in combination with lipids in the nervous tissues. For example, galactolipids. Okay, as a part of glycolipids. Okay, so that's about galactose. This fructose, galactose and mannose can be synthesized in the body from glucose. Okay, and when they metabolize also, when the fructose, galactose and mannose, when they metabolize, they can also form intermediates, which will ultimately enter into glycolysis. Okay, so that can be synthesized and they can be metabolized with products entering into glycolysis. Okay, fine. So what are reducing sugars? Reducing sugars are sugars with free anomeric carbon. Okay, because only when the anomeric carbon is free, they will retain the property of reducing some substances. Okay, if you go by the rule, all monosaccharides are reducing in nature. Okay, whereas sucrose, which is made up of glucose and fructose, is not reducing. I'll show you how. For example, you see here sucrose. It's made up of glucose and fructose. Okay, C1 is anomeric carbon of glucose. C2 is anomeric carbon of fructose. So both the anomeric carbons are involved in glycosidic linkage. So neither of the anomeric carbons are free. So that's why sucrose is a non-reducing sugar. Okay, whereas you take maltose. Maltose, galactose C1 and glucose C1, they are involved in linkage. Okay, they are free. Their functional group is free. So that's why lactose is reducing in nature. Maltose, glucose and glucose, they will be also reducing in nature. Okay, so if the functional group is free, then they are said to be reducing in nature. Okay, otherwise the anomeric carbon is free, then they can be involved in reducing the substances. That will give them the reducing property. Okay, so all monosaccharides are reducing sugars as their functional groups will be free, as their anomeric carbons will be free. Okay, fine. The classical test that is done in the labs, the first year medical practicals, is Benedict's test, which is the test for checking for reducing sugars. Okay, fine. Now let's go to now we have finished so far monosaccharides. Now we are going to disaccharides. Okay, disaccharides, it is made up of two monosaccharide units. Okay, monosaccharides are held together by glycosidic bond. Okay, sugars having free functional group, that is, if the functional group is not involved in the formation of glycosidic bond, then they are called as reducing sugars. We just now saw it. Okay, so first sugar is sucrose. Sucrose is a disaccharide which we commonly use in our day-to-day life. Okay, because our table sugar base is made up of sucrose. Okay, it is made up of glucose and fructose held together by alpha-1 beta-2 glycosidic linkage. And sucrose is metabolized by an enzyme called sucrase, which is present in the intestine. Very high levels of intake of sucrose can lead to dental caries. Then maltose and isomaltose. Okay, it is present in germinating cereals. Maltose and isomaltose, they are also made up of two glucose units. Okay, maltose is hydrolyzed by an enzyme called maltase. Isomaltose is hydrolyzed by an enzyme called isomaltase. Okay, fine. Now lactose. Lactose is present in milk and milk products, hence they are referred to as milk sugar. Okay, it is made up of glucose and galactose held together by a beta glycosidic bond, that is beta 1,4 linkage. Lactose can be synthesized in human, especially in the mammary gland during lactation. Okay, and the lactose which is present in the diet is broken down by lactase. Okay, the lactase is normally present in infants. However, as we age, we will tend to lose these enzymes. So that's why lactase levels will be low in elderly people. Okay, fine. So why it is important to know about this lactase? Okay, when the lactase enzyme is present, this acts on the lactose, which is made up of glucose and galactose, and break them into glucose and galactose. This lactase enzyme is present in the small intestine, especially in the membrane of the intestinal villi. Okay. What happens if the lactase enzyme is absent? The lactase enzyme is absent, this lactose cannot be digested. So what happens? The lactose will accumulate in the small intestine. And this undigested lactose is bad because they can be acted upon by the bacteria and they can release hydrogen, carbon dioxide, lactic acid and acetic acid. This hydrogen and carbon dioxide gas, so that leads to bloating, flatulence and abdominal pain. And this undigested lactose is osmotically active. So it can draw water from blood into the intestine and that can cause diarrhea. Okay. This will lead to a group of symptoms and that is characterized by a disease called lactose intolerance. Okay. This lactose intolerance is because of the deficiency of the enzyme called lactase. Okay. And these are the reasons why patients feel bloated, they have diarrhea, they'll have flatulence and they'll have abdominal pain. Okay. So the cause of lactase enzyme deficiency are only two things. One is age. As I told you, as age progresses, lactase levels will come down. Okay, so by 6 years of age itself, lactase enzymes levels will be very low. Okay, and this is common in Asians and Africans. Okay, second, if there is an intestinal disease, if the intestinal brush border is damaged, again lactase enzymes will be lost. Okay, so that is because of the secondary to intestinal diseases. Okay, fine. Next, the polysaccharides. Now we have seen monosaccharides. Now we have seen disaccharides. Now we are going to see polysaccharides. Okay. Polysaccharides are sugars that are made up of more than 10 monosaccharide units. They can be either homopolysaccharides or heteropolysaccharides. Okay. What are homopolysaccharides? Homopolysaccharides are those that are made up of repeating units of same sugar. It is homopolymer. The best examples are starch and glycogen. They are only made up of glucose. Okay. Then what are heteropolysaccharides? Heteropolysaccharides are polysaccharides that are made up of two or more different monosaccharide units. Examples: glycose, amino, glycans. Okay, it is also called as mucopolysaccharides. Okay, so that is homo and that is hetero. Okay, let's see the first homopolysaccharide, starch. The starch is considered as a major food reserve for plants. Okay, starch is stored in the cytoplasm of the plant cells as insoluble granules. What is starch made up of? Just now I told you. Starch is made up of only glucose molecules in repeating units. Okay. Structurally, starch is made up of two components, amylose and amylopectin. Okay. What is amylose? Amylose is a linear form of glucose residue held together by alpha-1,4 linkage. So this constitutes around 20 to 25% of starch. Okay. Amylose contributes to 20 to 25% of starch. Okay. Amylose is an unbranched form. It is an unbranched linear form. As you can see, it's linear. It's a linear form of glucose molecules linked together by alpha-1,4 linkage. Okay, and this contributes nearly 20 to 25% of starch. Okay, next, amylopectin. Amylopectin is a branched form. Okay, with alpha-1,6 glycosidic linkage. For example, so this is an amylose. Okay, and there's a branch here. And these branch points are seen in the starch at every 24 to 30 glucose residues. Okay. So it contributes nearly 75 to 80% of starch. Okay. So amylopectin is predominant when compared to amylose structure in a starch molecule. Okay, this is about starch. Okay, fine. Next we see about glycogen. Glycogen is also a storage polysaccharide, but it is found in animal cells. Okay. So it is stored in the cytoplasm as insoluble granules. Liver and muscle are the major sites of glycogen storage. Okay. It is a highly branched compound compared to starch, okay, with branch points at every 8 to 14 glucose residues, whereas in starch is around every 20 to 25. Whereas in glycogen, it's at every 8th glucose or every 14th glucose there will be a branch. Okay, that's why glycogen is also called as animal starch. Okay, fine. See here, see how highly branched starch glucose is when compared to starch. Okay, so there's a branch point at every 8 to 14 glucose residues. Okay, fine. Now let's go to the cellulose. Cellulose is a homopolysaccharide. It is not a storage polysaccharide. It is a structural polysaccharide. It is a homopolymer of glucose held together by beta-1,4 glycosidic linkage. So in starch and glycogen, they are made up of amylose and amylopectin. Amylose by alpha-1,4, amylopectin by alpha-1,6. In cellulose, they are made up of glucose, but they are held together by beta-1,4 linkage. Okay, it's a primary structural component of the plant cell wall. Okay, do we have the capacity to digest this? No, we do not have an enzyme called cellulase to digest this. So cellulose is indigestible by humans. Okay, fine. So cellulose is one of the important components of dietary fibers, but we are advised to take dietary fibers because it has a lot of other health benefits. Okay, it reduces cholesterol, it increases the glycemic control, and then it prevents constipation, it adds roughage to the stool. That's why our food should always contain dietary fiber. Okay, fine. Now the next one, inulin. Inulin is also a polysaccharide. It is a homopolymer of fructose, okay, not glucose. Okay, whatever you have seen so far, starch, glycogen, and cellulose, they are homopolymer of glucose, whereas inulin is a polymer of fructose. It is found in the tubers of dahlias, artichokes, and dandelions. Okay, it is readily soluble in water, not hydrolyzed by the intestinal enzymes excreted in urine. We do not have enzymes to metabolize inulin. So if you take inulin, it will come out as inulin in the urine. Okay, this property is utilized in estimating glomerular filtration rate by using inulin. Okay, which is a marker for kidney function. Okay, but inulin is not used nowadays to estimate glomerular filtration rate, but that is a gold standard technique to measure GFR. Okay, fine. Okay, next, mucopolysaccharides. Okay, we have seen homopolysaccharides, glycogen, starch, cellulose, inulin. Now we are going to see mucopolysaccharides. Okay, mucopolysaccharide is a heteropolysaccharide. Okay, it is an unbranched molecule made up of repeating units of two substances. What are two substances? Uronic acid and amino sugar. That's why they are also called as glycosaminoglycans. Okay, and these glycosaminoglycans are produced in endoplasmic reticulum and Golgi apparatus by a complex process. Okay, fine. So this is a repeating units of uronic acid and amino sugar. Okay, fine. Let us see in detail now. Okay, uronic acid is nothing but sugars in which the terminal carbon's hydroxyl group, okay, undergoes oxidation to form carboxylic acid. Examples are glucuronic acid and iduronic acid. Okay, for example, this is glucose, C1, C2, C3, C4, C5, C6. Okay, when this hydroxyl group on the C6th position undergoes hydroxylation to form COOH, then that becomes glucuronic acid. Okay, and this is iduronic acid. Okay, one is glucuronic acid, the other one is iduronic acid. Okay, what's the difference between these two? Just see the position of H and OH groups at the C5th position. See here, OH group is on the right side, but here it is on the left side. That's the difference. Okay, so that is the difference between glucuronic acid and iduronic acid. How glucuronic acid is formed? By the hydroxylation of OH group of the oxidation of OH group at the terminal carbon atom. Okay, that is glucuronic acid and iduronic acid. Okay, so as I told you earlier, glycosaminoglycans are unbranched molecules made up of repeating units of uronic acid and amino sugar. So what are amino sugars? Can be either glucose amine or it can be galactose amine. Okay, so this is glucose amine. See here, amine group at the second carbon, glucose amine. Okay, and these amino sugars exist in the acetylated form. That is, they will exist as N-acetyl glucosamine or N-acetyl galactosamine. So this is N-acetyl glucosamine. Okay, so amino sugars are glucosamine and galactosamine. These amino sugars usually exist as acetylated form, N-acetyl glucosamine or N-acetyl galactosamine. Okay, fine. So these glycosaminoglycans are negatively charged molecules. They tend to repel each other. Okay, and they are surrounded by shell of water molecules. Okay, so when you put glycosaminoglycans together in a solution, that solution becomes highly viscous. The solution will become elastic, resulting in mucus-like consistency. That's why these glycosaminoglycans are also called as mucopolysaccharides. Okay, so this figure will explain you the viscous-like consistency and their lubricating effect of glycosaminoglycans. Okay, these glycosaminoglycans, because of this negative charge, as I told you earlier, they will try to take water in them, like this. See here, the red color which you see is the glycosaminoglycan chain, and they will be bound with water. When you compress this, the water molecules will come out. When you release the compression, the water molecules will again go inside. So that's a cushion, right? Cushion is when you press it, it will compress. When you release it, it will come back to its original position. Okay. So glycosaminoglycan also contributes to this cushioning effect because of this property of taking water inside and letting the water outside. And that is because of the negative charge conferred by the amino sugars and uronic acid. Okay, fine. So that's about the glycosaminoglycans. Okay, these glycosaminoglycans are found in various parts of our body. They are found in synovial fluid of the joints, vitreous humor of the eye, skin, and blood vessels. The property of the glycosaminoglycans to get adjusted to pressure and friction contributes to the resilience of the synovial fluid or the vitreous humor of the eye. See, these glycosaminoglycans are found embedded in the ground substances of extracellular matrix. Okay, see, this is an extracellular matrix. So it's an epithelial layer. These are other cells in the extracellular matrix, and there is some ground substance. And this ground substance is made up of glycosaminoglycans, along with proteins like elastin and collagen. So which is the ground substance of extracellular matrix? It's mainly composed of glycosaminoglycans. So these are the 6 types of mucopolysaccharides that is found in our body. Hyaluronic acid, heparin, heparin sulfate, keratan sulfate, dermatan sulfate, chondroitin sulfate. So these are the 6 glycosaminoglycan molecules. Okay. So here, this table will tell you the name of the glycosaminoglycan molecule, where it is found, and what are the components. Okay. Hyaluronic acid is present in the extracellular matrix. Okay. Just now I told you, it contributes to the ground substance of the extracellular matrix, especially in the bone, cartilage, joints, and vitreous humor. Okay. It is made up of glucuronic acid and N-acetylcosamine. So coming back to the basic definition of glycosaminoglycans, they are made up of uronic acid and amino sugars. Chondroitin sulfate is found in the cartilage. They are also made up of glucuronic acid, but they are made up of N-acetylgalactosamine. Keratan sulfate, they are found in the cornea. They are made up of galactose and N-acetylglucosamine. Okay. This keratan sulfate, which is found in the cornea, is responsible for maintaining the transparency of the cornea. This is a very important NEET PG and INECET question. Okay. Which is responsible for maintaining the transparency of the cornea? Keratan sulfate. Then dermatan sulfate, which is found, as the name suggests, dermatan. It is found in the skin. This is made up of uronic acid and N-acetylgalactosamine. Heparin, which is an anticoagulant produced by mast cells, liver, lung, and skin. They are made up of either glucuronic or uronic acid, and that is made up of only glucosamine. Okay. Heparin sulfate, basement membrane, glucuronic acid, and glucosamine. So these are the various glycosaminoglycans, their location and their components. Now we are going to see the functions of glycosaminoglycans. Okay. I told you earlier itself, it is a ground substance of extracellular matrix. Second, it helps in maintaining the elasticity of the tissue. It mediates cellular migration. Okay. It helps in maintaining the transparency of the cornea. It acts as an anticoagulant. They are the components of cell membrane. Okay. Fine. Heparin, it is produced by the mast cells that line the arteries, capillaries, especially in the lung, liver, and skin. It acts as an anticoagulant. That's why heparin is used in the treatment of myocardial infarction and stroke. The enzyme hyaluronidase, which breaks the glycosaminoglycan, is secreted by the cancer cells, so that the cell-to-cell connection is lost and the cells can migrate from one place to another place, which is called metastasis. Okay. Again, this hyaluronidase is also used as a drug to treat a condition called frozen shoulder. Okay. So they give this drug into the joint. It will go and cleave this hyaluronic acid thing, and it will free up the shoulder. Okay. So that is one more property of this hyaluronidase. Okay, fine. This, as I told you earlier, these glycosaminoglycans are produced in the cell, especially in the endoplasmic reticulum and Golgi bodies by a complex process. Okay, and they are degraded in the cell in the lysosomes by specific enzymes in the lysosomes. Okay, so any defect in those enzymes would lead to accumulation of glycosaminoglycans inside these cells. So this causes damage to the tissue, especially the lungs, liver, brain, bone, and kidney. And this condition is called as Mucopolysaccharidosis. Okay. So, defect in the lysosomal enzymes involved in the breaking down of glycosaminoglycans can lead to accumulation of glycosaminoglycans, and it can lead to a condition called mucopolysaccharidosis. Okay. So, mucopolysaccharidosis can affect any of the following organs. For example, if it accumulates in the brain, that can lead to mental retardation. If it affects the cornea, that can affect the transparency of the cornea. If it affects the liver, it can cause hepatomegaly. Okay. If it affects the spleen, it can cause splenomegaly. Okay. If it involves a bone, it can cause bone deformity. So these are the certain examples of mucopolysaccharidosis. There's something called Hurler's disease. There's something called Hunter's disease. There's something called Sanfilippo's disease. These are the 3 diseases, major diseases of mucopolysaccharidosis. Okay. For example, Hurler's disease is caused due to the defect in the enzyme called L-iduronidase. When this enzyme is defective, it leads to accumulation of dermatan sulfate and heparan sulfate. Hunter's disease caused due to the enzyme called iduronate sulfatase. That leads to accumulation of both dermatan and heparan sulfate. Sanfilippo is caused due to the defect in this enzyme, N-acetyl glucose aminidase or glucose aminide acetyl transferase or glucosamine sulfatase. Accumulation of heparan sulfate. Morquio disease, accumulation of keratan sulfate due to the defect in the enzyme called galactose aminidase or galactosamine 6 sulfatase. Okay. So these are the 4 mucopolysaccharidoses. These are the enzymes involved and these are the metabolites that can get accumulated. Okay, fine. So if the glycosaminoglycans deposits in the brain, it can lead to mental retardation. If it deposits in the liver and spleen, it can lead to liver enlargement and spleen enlargement. Okay. If keratan sulfate is not there, keratan sulfate is responsible for maintaining the transparency of the cornea. If keratan sulfate is not there, okay, then the cornea will become clouded. Okay. It involves the joints. Joints can become stiff. That's called joint contractures. Okay. And there will be failure to thrive and there will be leukocyte inclusions. Because of the lysosomal enzyme defect, this cannot be degraded. So they will form some inclusions in the lysosomes. That will be reflected as leukocyte inclusions when you see under the microscope. Okay. One important finding or one important question that was asked repeatedly in the previous year questions were, they will give you a case history with mental retardation, liver and spleen enlargement, everything. If you see corneal clouding, that is highly unlikely to be Hunter's disease because corneal clouding is not seen in those with Hunter's disease. How can you remember this? Hunters should have a clear eye, so they cannot afford to have a clouded cornea. So corneal clouding is not seen in those with Hunter's disease. Okay. However, it is seen in those with Hurler's disease, Sanfilippo and Morquio's disease. Okay, fine. So that's about this condition. Okay. Now, that's about glycosaminoglycan. Okay. There's one more compound called proteoglycan. What is it? This proteoglycan is nothing but the glycosaminoglycan chains attached to a protein. Okay. So for example, you see here in this figure, what you see as a green is a protein. And what you see as this black thread-like structures, they are nothing but glycosaminoglycans. So glycosaminoglycans attached to a protein, they are called as proteoglycans. All glycosaminoglycans except hyaluronic acid exist in the form of proteoglycans. Okay, because hyaluronic acid will be acting as a link for this two proteoglycan monomers. Okay, so that's about proteoglycans. Proteoglycans are not synonymous with glycosaminoglycans. Both are different. Okay, so glycosaminoglycans in chains attached to a protein, that is called proteoglycans. Almost all glycosaminoglycans except hyaluronic acid will exist in the form of proteoglycans. Okay, then what is glycoprotein? That is even more different. That is still different from what you have seen so far. Glycoproteins are proteins to which oligosaccharide chains are covalently attached. Where have you seen this? We just now discussed. Glycoproteins are found in the plasma membrane. Okay, so length of the carbohydrate chains attached to a protein is short compared to those in the proteoglycan chain. Since membrane-bound glycoproteins have wide variety of functions. For example, they are responsible for cell surface recognition. They act as receptors. Okay, they also act as blood group antigens and immunoglobulins. Okay, so glycosaminoglycan is not synonymous with proteoglycan, and proteoglycan is not synonymous with glycoprotein. Okay, glycoprotein is just a protein attached to an oligosaccharide chain, which is found along the plasma membrane. Okay, fine. They can act as receptors, they can act as blood group antigens and immunoglobulins. Okay, so with this we have finished the chemistry of carbohydrates. So we have seen how carbohydrates are classified as mono, di, poly and oligosaccharides. We have seen the examples of monosaccharides, disaccharides. In disaccharides also see something called lactose intolerance. Then we saw polysaccharides, which is homopolysaccharide and heteropolysaccharide. We saw about starch, glycogen, cellulose, insulin. Then hetero we saw about something called glycosaminoglycan, proteoglycan and glycoprotein. We saw a group of disorders called Mucopolysaccharidoses. Okay, with this session will be completed. Thank you.